Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss a topic called asexual reproduction in bacteria. So bacteria can do asexual reproduction by the following methods. They are binary fission, endospore formation, gonidia formation, bud formation, then by cyst formation, they can do asexual reproduction. So in this video, we are going to discuss the asexual reproduction of bacteria in detail. First, let me explain the process binary fission. So binary fission is nothing but in which a single cell of bacteria can be divided into two equal daughter cells, and that process is called as binary fission, and it is the most common method of bacterial multiplication during favorable condition. Then let me tell you the steps which are involved in binary fission. So this is a bacterial cell. So it is a E. coli bacteria which contains the genetic material nuclei. Nuclei means they do not possess a nuclear membrane and they are not well defined nucleus. That is why they are called as nuclei, and the genetic material is a double-stranded circular DNA. So E. coli contains double-stranded circular DNA. So the double-stranded circular DNA undergoes a process called replication. Replication is nothing but in that process, this double-stranded circular DNA going to make an exact copy. So the double-stranded circular DNA undergoes the replication process. During that replication process, the double-stranded circular DNA get separated. So this is the original strand of DNA. So this is the one. This is one strand, and this is the another strand. This is also original strand of the bacteria. Then during replication process, the complementary strands are produced on the original strands. So during replication process, complementary strands are produced on the original strand. Then now you can find two identical double-stranded DNA. Then these two identical double-stranded DNA are distributed to poles of the dividing cells. So this is the two identical double-stranded DNA, and they are distributed to the poles of dividing cells. Then here you have to notice a point. Here there is no formation of spindle formation. There is no formation of spindle fibers like mitotic cell division. So here you are not supposed to observe the formation of spindle fibers which happens in mitotic cell division. So that is the major difference between binary fission and mitotic cell division. Then a septum. This is a cross wall. So a septum or a constriction which takes place in the middle region. Then the cells get separated into two equal daughter cells. So that's all about the binary fission process. Then I am going to tell about gonidia formation by the bacteria. Gonidia formation takes place in filamentous bacteria. Filamentous bacteria are nothing but they contain a long strand. So this is the long strand. So a bacteria which contains a long strand body, then bacteria are called as filamentous bacteria. Then you can find the gonidia which are formed at the apex of the filament. So these are all gonidia which are formed at the apex, that is the tip of the filament. So the gonidia are formed at the apex of the filament. So the filament which bears the gonidia is called as gonidiospore, which is called as gonidiospore. Gonidiospore. So this is a filament which bears gonidia is called as gonidiospore. Then this gonidia can separate from the mother bacterial cell, and getting suitable substratum, and it can get suitable substratum and it can develop into a new bacterial mycelium. Here I have point a term called mycelium. Mycelium means the term which are used generally in the fungi. Generally we are using the term mycelium in fungi, but here I am using mycelium because this is a bacteria which shows the bacterial body which looks like fungi. That is why I use the term mycelium. Actually that bacterial class is called as actinomycetes. In that class of bacteria, the bacterial body shows or resembles the fungi body. That is why they are called as actinomycetes. So the gonidia can detach from the mother and it can get the suitable substratum. Then it can develop into a new mycelium. Example for gonidia formation: streptomyces. Then I am going to explain the bud formation. So here you can find a bacteria cell which develops a swelling. So this is a bacterial cell which develops a swelling. First it looks smaller in size. In later it gradually increase in their size. First you can find it is smaller in size. Later it can increase their size gradually. Simultaneously nuclear division also takes place inside that cell. So one part of nucleus remains with the mother cell, one part of the nucleus remains in the bacterial cell, that is the mother cell. Then other part of the nucleus goes to the cytoplasm of the bud. So once again I repeat the statement: one part of the nucleus remains within the mother cell, that other part of the nucleus goes with cytoplasm of the bud. After getting detached from the mother cell during favorable condition, that bud can develop into a bacteria. During favorable condition, this detached bud can develop into a new bacteria, and that formation is called as bud formation or budding bacterium. For example, for budding bacterium is rhodomicrobium. So example for budding bacterium is rhodomicrobium. Next I am going to tell about cyst forming bacteria or cyst formation in bacteria. So cyst formation is nothing but formation of additional layer around the mother cell. So cyst formation is nothing but formation of the additional layer around the mother cell is called as cyst formation. Generally the cyst formation takes place during unfavorable condition. So the bacteria remains as such. They do not do any metabolism and they do not multiply at that time. So at that time only the cyst formation takes place. During favorable condition, the bacteria can behave as a mother cell. The bacteria can behave as a mother cell and it can multiply into many bacteria, and that is called cyst formation. For example, the cyst forming bacteria Acetobacter. So example for cyst forming bacteria is Acetobacter. And in some aspect, the cyst resembles the endospore. However, the structure of cyst and their endospore is totally different, because endospore can withstand high amount of heat. The endospore can resistant to high amount of heat. Then I am going to tell about the endospore formation in bacteria. So during unfavorable condition, the bacterial cell develop this endospore. So the spore which develop inside the bacterial cell. So this is a spore which develop inside the bacterial cell. That is why they are called as endospore. So this endospore can prevent the bacterial cell from unfavorable condition like desiccation and starvation. Desiccation means this endospore prevents the bacterial cell from the dryness, and starvation. Then the example for endospore formation bacteria are Bacillus megatherium, then Bacillus sphaericus, then Clostridium tetani, So these are all examples of endospore formation bacteria. Then the endospore can resistant to high amount of temperature up to 100 degree Celsius. So the endospore can withstand up to the temperature 100 degree Celsius, and it can resistant to toxic chemicals. So the resistant nature of the endospore is due to the presence of thick wall. So the resistance of the endospore is due to the presence of thick wall. Then the thick wall contains low amount of water. Then it contains a chemical called gyplonic acid. So these are the reasons that endospore can withstand high temperature and toxic chemicals. Then that bacteria which produce this type of endospores are not able to kill by antibiotics or any sterilization method, because it can withstand any kind of condition. It can withstand high temperature and it can withstand the toxic chemicals. Fortunately, the pathogenic bacteria which produce this endospores are only two bacteria. So two pathogenic bacteria can produce this type of endospore which can cause disease, and that bacteria are called as anthrax and tetanus. So these two are very serious disease which is caused by the bacteria which can produce endospore. So these two pathogenic bacteria can produce endospore. So we have to be very careful with this kind of bacteria, because it contains endospore. It cannot easily killed by antibiotics and heat. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.